The Honorable chemistry student and the participant of the guest lecture. Alhamdulillah, thanks to His grace and guidance, so we can gather in this room in a good health. We also don't forget to convey prayers and greeting to the great Prophet Muhammad Sallallahu Alaihi Wasallam. Starting today's agenda with Bismillahirrahmanirrahim. Ladies and gentlemen, the next agenda, welcoming speech from the coordinator of Chemistry Study Program, Mr. Indra Lesmana Tarigan, SPD NSC. Please, time is yours.
Okay, thank you, Sylvina. Good morning from Hira. It's pleasure that praise and gratitude to Allah Subhanahu wa Ta'ala for presence and blessing, and also for distinguished lecture in Department of Chemistry, Faculty Science of Technology, Science and Technology, University Jambi. And we from the part of Department of Chemistry would like to express our gratitude to Associate Professor Hira Khalid, who is able to attend our guest lecture today, and actually also for all of our students who have attended this guest lecture activity. And in this a special moment for us to have a guest lecture from the Forman Christian College University, Pakistan, Associate Professor Dr. Hira. She is an expert in chemistry, especially in organic chemistry, drug discovery from the Pakistan, and also a member of Royal Society of Chemistry, and also American Chemical Society. And our moderator will later introduce her in more detail. And we need to inform to Dr. Hira, actually, regularly we invite the chemical expertise from within the in the country, and also from the abroad, international guest lecture every month to build an academic culture, academic atmosphere in our department. Also in this context, for the internationalization of our department, we also have one current student, first year student from Pakistan. I think he was present later also in this guest lecture. And more than that, we would like to thanks again to Dr. Hira. I hope this is a good initial communication and collaboration. And in the future, we hope we can build various field cooperation, like the academic collaboration, research collaboration, joint supervisor, and other activities that we can initiate. And this activity initially and officially we open by saying Bismillahirrahmanirrahim, and we begin today's our guest lecture activities. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Assalam, and thank you so much, Sir Indra, and for your kind invitation. And it is always delighted for me to interact with the Indonesian faculty and the students. I taught one of the course in the Bangalore University, and now this is the opportunity that I'm collaborating with you in a Jambi University. So I really appreciate. I just see some of the the videos you have played, at whatever the efforts you are making for the international collaboration and improving and uplifting your research and your academia, especially the teaching and the learning. So thank you so much for the invitation, and I'm delighted to be there. So before starting, let me share my presentation. So I hope I will be in the time. So I just wanted to make sure that you are able to see the slides of
yes. But before you give speech today, we have our moderator today is our former student. He was graduate from the Pennsylvania University, and he will guide our guest lecture today. I mean, and he will introduce you more detail and present your short CV.
Okay, sure,
Okay,
sure.
Sylvina.
Thank you, Mr. Indra Lasmana Tarigan. The next schedule is the reading of the prayer, which will guide by Rifat Choki. Please, time is yours.
Assalamualaikum warahmatullahi wabarakatuh. A'udzubillahi minasyaitonirrajim. Bismillahirrahmanirrahim. Alhamdulillahirabbil alamin. Hamdan yu'afi ni'amahu wa yukafi mazida. Ya Rabbana lakal hamdu wal kasyukur kama yanbaghi li jalali wajhikal karimi azimi sultanik. Allahumma sholli ala sayyidina Muhammad wa ala ali sayyidina Muhammad. Rabbana hab lana min zurriyyatina qurrata a'yun waj'alna lil muttaqina imama. Rabbana la tu'akhizna in nasina aw akhtana. Rabbana wa la tahmil alaina isran kama hamaltahu ala allazina min qablina. Rabbana wa la tuhammilna ma la taqata lana bi. Wa'fu anna waghfir lana warhamna. Anta maulana fansurna 'alal qaumil kafirin. Allahumma inna nas'aluka salamatan fid dunya wal akhirah wa afiyatan fil jasad wa ziyadatan fil ilmina wa barakatan fil rizqina wa taubatan qabla maut wa maghfiratan ba'da maut wa najatan minan nar wa afain dal hisab. Ya Allah, bless all the knowledge that we get during the activity so that we can practice it later. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina azabannar. Wa sallallahu ala sayyidina Muhammadin wa ala alihi washabihi wasallam. Subhana rabbika rabbil izzati amma yasifun wa salamun alal mursalin.
With the reading of this prayer, the opening session of guest lecture in this morning is finished. Hopefully, this activity can run smoothly until the end. Amin. Allahumma amin. The next agenda is group photo session for documentation commit. Please take the picture. One, two, three. The next slide. One, two, three. The next slide. One, two, three. Okay, I'm Sylvina Fradila as Master of Ceremony. Apologize for my mistake in delivery this opening event. And I think that's all. Wassalamualaikum warahmatullahi wabarakatuh. For the next to the next moderator, please, time is yours.
Right. Thank you so much, Sylvina, for the time and place. Yes, first I would like to say welcome to Dr. Hira Khalid today in our meeting. This is very delightful and happy experience to us to welcome you and also having you here as our guest lecturer in this moment. First, I would like to introduce myself before I introduce Dr. Hira Khalid, of course. So my name is Muhammad Hira Salihin, usually called Irhas. I'm just graduated from University of Pennsylvania, United States of America, in organic chemistry. Okay, that's my short introduction. Now let me introduce Dr. Hira Khalid before she presents the presentation. Okay, we know that Dr. Hira Khalid right now associates with Furman Christian College University, Pakistan. She is a distinguished professor with qualifications. She was a U.S. Fulbright postdoc at University of Minnesota, Twin City, Minnesota, USA. With the education background, she got her bachelor and master degree from University of the Punjab, Lahore, Pakistan, and the PhD degree from University of Lahore, Pakistan. Her experience, the recent one was she was U.S. Fulbright visiting professor. That's really amazing. Hopefully that we also can follow her step. I mentioned before that the visiting professor was conducted at University of Minnesota, Minnesota, USA. Also, she is associate professor now in Furman Christian College University, and was assistant professor at Furman Christian College University in Lahore, Pakistan, too, from January 2017 to April 2020. And so much experience, sure, that I could mention one by one, but they are all amazing. Sure, she teaches some course right now at the university, such as Organic Chemistry 1, Organic Chemistry 2, Biochemistry, and related field. For the research projects, she has some projects right now currently active and also completed project. One of the current project that is in progress is with title In Vitro and In Vitro Pharmacological Cytotoxic Studies of Sulfonamide Derivatives of Piperidine. She has a lot of activities and memberships as well during her career. Currently, she is an advisor in ACS Punjab chapter Pakistan, and also a U.S. Fulbright Association, and a member of PULSE, Partnership of Undergraduate Life Science Education, and many more. She has so many expertise in instrumental chemistry that I could mention one by one, but some of them are NMR, dry box, and atomic absorption spectroscopy, UV spectrophotometer, etc. And that's what I could mention right now. Sure that there's still a lot of things that to mention about Professor Dr. Hira Khalid. She is a distinguished professor from Pakistan. Now, place to Professor Dr. Hira Khalid. Place and time are yours.
Thank you. Thank you so much, Mohammed Irhash. Thank you for that overwhelming introduction, and I'm really humbled for that. So now, starting about the discussion, let me, if I could get the opportunity to share my screen, so then I can share. Yeah, thank you. Okay, so I hope everybody is able to see the slides. So just wanted to make sure if anybody can respond that you are able to see that one, so I can start further.
Yes, we could see the slides.
Okay, thank you for confirming. Okay, so as the theme is about this international guest lectureship, and I was conveyed by the organizers that I should talk about the drug discovery, particularly with the focus on the organic synthesis. So I plan my talk regarding this one because I also always get inspired about the drug discovery and the design. So basically, when I did my MS in analytical chemistry, so organic lab was always something much interesting, magical for me because you create something, you're doing the elemental testing, element or the group functional group detections. So it always inspired me. So I switched from the analytical chemistry to the more on the synthetic side. So then I shifted my interest to the medicinal chemistry, where I added the computational organic synthesis, and this is also the interdisciplinary, the cross-discipline area where we have the information and the knowledge, and we utilize our knowledge of the organic synthesis, biochemistry, and the computational chemistry. So my aim for today is that I could not only give you the idea what the whole of the drug discovery journey is, but I will also give you the insight that how a chemist is able to plan a project and what are the things we needed for doing any of the work or designing any of the project when we are interested for designing and the discovery of the drug, and especially when we are with the organic or the synthetic chemist. So what are the approaches we utilize for in the discovery of a drug? And secondly, that you would be able to compare that either this drug discovery is an easy or the challenging. So what are the challenges and where we are as a chemist playing our role. So I took the example from one of the projects from my research group. So my group is always interested for designing the small molecule based on the heterocyclic compounds and then their biological potential. And we design the compounds according to whatever the target we decided and what disease we are interested in to combat that one or to provide some of the solution to treat in the future. So my today's topic is under the theme of the drug design and the discovery is the synthesis of 1,2,4-triazole derivatives as potential acetylcholinesterase inhibitors. So I will give you the introduction as well, that what the acetylcholinesterase is and why we need the inhibitors and what is the importance of these enzymes. So firstly, before going into the detail that whatever the work we have done and how we design one of the project and we synthesize some of the compounds and then we got some of the good results that it can help for curing the disease, the Alzheimer. But I wanted to give you the idea that what is the whole of the journey when we talk about the drug discovery, right? So if you just look at this slide, so the drug discovery is the first step in the whole of the journey when you get the tablet in your hand for the treating any of the disease. So the first part is the drug discovery, the second part is the clinical testing, and the third part is the approval for production. And these all three steps, you can say, or all three phases in the drug discovery are very important. So the major part where we are contributing as a chemist is basically this yellow part, which is called as a drug discovery. So we being a chemist and either we pharmacists or the biochemist or the biophysical scientist. So we are largely contributing into the drug discovery because we start from the scratch to this one. Now, the important thing is when there was a pandemic, when there was a COVID, so many of the people have this question in their mind and they were expecting that there should be the drug in the one night. There are many of the chemists, there are many of the scientists, There are so many huge and the big names of the pharmaceutical industries, and they should come up with a solution in a one day. But the thing is, when you are in a scientist, when you are in a field, and you really have the idea that how this journey do happen, so this is not a one night solution. It demands a lot of time, effort, and the money. So obviously, I cannot do like if I do the reaction and I create some of the compounds, and even if it is, I'm thinking that could be the biochemically active or biologically active, I cannot directly give it to somebody, not even animals, not even the human being. So there are many of the studies which have to be done. So that require the toxicity, that require the purity, that require that how much we should set the dose for that one. So that's why if you just look at this one, so the very first thing is in the drug discovery, as a chemist, if you wanted to work on something, so the first thing is the target identification. So for example, the example I'm giving you today is that I wanted to target on the treatment of the Alzheimer, because Alzheimer is also one of the biggest challenge. And still the scientists are working on proposing the more better, the more efficient drugs for that one. So before, when you decide that you wanted to go into that field and you wanted to work on this particular disease, that could be anything. That could be the cancer, that could be any particular cancer, that could be the any of the infectious diseases. But keeping our example or a presentation story in the mind. So if I wanted to work on the treatment for the Alzheimer, so the first thing is that I have to identify the target. So what the target identification is, that there is particular protein which is responsible for that major function. So either that function in this response of that disease, either this is possible that that particular protein or the enzyme is not active anymore, or this is overactivated. So the first thing is to find the structure of that protein, right? And then we go for many of the options. So for example, this lead discovery, this lead optimization include that once we have the protein, then the scientists go for their crystal structure, that if they have the clear exact structure of that protein, that what are the amino acids which are making that protein, and what is the active site of that one, which site you can target and where your molecule can bind to that one, and then either that can stop its activity or that can enhance its activity. That depending, because most some of the diseases do happen because of the overexpression of any of the enzyme, and some are happening because of the suppression of that enzyme activity. So in the lead discovery, here we utilize our organic chemistry knowledge, that what should be that compound structure. So there are many of the approaches. For example, the first approach is that you did the literature review, and there are the papers, the publications, that they are suggesting that this kind of molecule and its structure is showing some of the good results, and they are using in the treatment for this disease, this kind of disease, but it has some of the side effects, or maybe it is not has been synthesized in the labs, but this is available in the plant sources. So then the scientists do through the literature review, and another important thing, which comes with some of the last few years, and which has enabled the life of the scientists, and especially who are working in the medicinal chemistry and for the drug designing, this is the computational tools. So these are basically the computer based, as now this is a time everyone is talking about the AI. So these are the predictive exercise that you screen whatever the compounds are available, whatever the drugs are available. And sometimes we do that, we provide our protein structure, and then through the computational tools, be able to analyze that that should be our ideal compound, which can bind from these amino acid residues, and that can show some of the activities. So through utilizing the computational tools, we get, or from the literature, we get an ideal structure or an idea or predictive structure that should be our model structure of the compound. Then the chemists do synthesize in their lab, and then after synthesizing any of the compound, you also have studied in your courses as well. So when we synthesize something, so there is the important thing is that we have to optimize that reaction, that we can reproduce that one. Second thing is that we have to characterize that one, that either that is the actual compound, we are successful to make that one, or there is this is not the same compound we were aiming to synthesize. So how to confirm that one? So there are many of the techniques and more of the advanced one, and some of which we also utilize in our labs as well. In our teaching labs, we study their theories as well. So the most significant one are the NMR. There are the X-ray if your compound is the crystalline one. So after doing the characterization, so these compounds then firstly go for the in vitro biological assays. And we use the cell lines, we use the proteins, or we use the enzyme testing accordingly, that whatever your target is that you wanted to see the behavior of your compound against that one. So then after in vitro analysis, if your compound, say, has shown some of the good results, so then we go for the preclinical testing. And the preclinical testing is something when you get the compound with the purity of 95, above 95%. So we don't compromise the animals if our compound's purity is less than the 95%, because you have to give that compound which has that much pure, so then you can do the exact studies on the animals. So after the success from the animal testing, then we go for the phase 1, phase 2, and phase 3. These are called the clinical trials and the clinical testing. So let's say, and just if you just see in the slide below, so this whole of the process, which is the yellow one, which is a drug discovery, it exercises, and it costs $600 million to $800 million, and this is some of the standard or the minimal prices. So that can exceed then that one, depending on that if you are facing many of the challenging way in the drug discovery. So usually what happens that after doing many of the series of the compounds, the chemists are working, they are doing their biological assays, they are doing their animal testing. So they are usually the 200, let's say in initial, we have the 200 compounds we have synthesized, and that goes to the preclinical testing. Then in the next phase, this demands 6 to 12 years because there are the different trials in a phase 1, whatever, either there are some of the toxicity reported or not, then they proceed for the second and then the third one. So it usually demands 6 to 12 years. And this costs $1.2 to $1.4 billion. And from the 200 compounds, we shorten to the 5 entries. Why? Because usually it is a usual score. So we make the different compounds, but not all of the compounds are active. So sometimes it shows that they could have the activity, but when they go in the real animal system, in the cellular system, so they behave differently. So then we just left with the 5 entries. 5 entries mean that we just get the 5 compounds which are showing the promising thing after the preclinical testing. And then at the last, let's say from the 200, we initially synthesize it, then we get only the 1 compound which has passed all of these steps. So the regulation, the approval, it demands 1 to 2 years, and there is the Food and Drug Authority. And some of the countries also have their own regulatory bodies. So they do and it demands almost a $50 million. So only the one drug gets from all of these as FDA approved, and once that FDA approved, so then that drug is able to market and to suggest to the patients, because now this compound has been set for the dose, that how much the minimum amount we have to give. For example, in drugs, you have seen that there are some of the drugs the doctor prescribes you. These are the 5 mg, the 50 mg, or depending on their doses. So these all things happen during the clinical testing and the approval of the production. So my aim was this one, that you should realize that this is the whole of the process, and this is really not an easy task. So there are many of the scientists, their efforts, their years, and a great amount of expenses are basically involved for the drug discovery. So now moving towards our major main topic. So we are covering today one of the heterocyclic moiety today, and that is the 1,2,4-triazole heterocyclic compound. So the heterocyclic compounds are, they always inspire me as well, because I have seen in many of the phytochemicals, the plant-based compounds we are using for in many of our conventional treatment as our the natural prescriptions. So then I got the interest in the nitrogen-based heterocyclic compound. So the nitrogen is an amazing element because of its electronegativity, because of its 3-bond formation capacity, and the lone pair of electron. So nitrogen-based heterocyclic compounds have the tremendous applications, and because of their diverse role. For example, you can see the alkaloids, you can see in many of our important ingredients in the spices we use in our continents. So the black pepper, piperidine, the pyrrolidine, the pyridine. So you have seen many of the nitrogen-based compounds, and alkaloids, major of the class of the nitrogen-based compounds. You also see the amino acids. They have the nitrogen in their structures, but all are the heterocyclic compounds. Had a wide spectrum of the biological activity that includes antifungal, antibacterial, analgesic, anti-cancer, antidepressant, anti-HIV therapy. They are also useful in many of the drugs which treat the antiviral diseases, anti-TB, anti-inflammatory, and antioxidant. So they all are the important, making the important class of the compounds which are have application in medicinal chemistry. So now if we switch to the next, so what the 1,2,4-triazole is. So the triazole is a 5-membered 3-nitrogen containing scaffold, and the heterocyclic compounds has a different pattern of naming. These are under the IUPAC accepted rules, but these are not following the similar kind of pattern of the naming as we do the other organic compounds. So for the simple idea to you, so whenever we do have the 5-membered ring and they are the saturated one, so we use OLE at the end. So this is representing that your given structure is a 5-membered ring, and it has the unsaturation, it has the aromaticity. Then the tri means this is the 3, and aza is a one of the descriptive name or the term. For example, the methyl, ethyl, propyl we use. So the aza is basically to representing the heteroatom which is in nitrogen. So if there's a nitrogen, we will use aza. If there will be the oxygen, we will use the oxo, and etc. So the 1,2,4 is representing that in this 5-membered ring, so where these nitrogen is. So if you just look all of these 3 structures, so they have the 1, 2, and the 4. So they have arranged in that one. So this is important to mention. There are other isomers as well. So that could be the 1,3 1, 2, 3, that could be the different one. So this particularly, I'm talking about the 1, 2, 4 triazole. If you just look at this slide, so I have presented the 3 structures. So maybe these are a little bit scary to you, that these are 2 complex structures. So these are basically these 3 drugs which are available in the market. And the important is that I wanted to emphasize that what is an importance of the 1, 2, 4 triazole heterocyclic compounds. So the first one, which is the triazolam. So this is used for the insomnia, which is one of the sleeping illness, to treat that one. The next one is the aprazolam, and this is for the treatment of the anxiety and the panic disorder. The third one is the muscle relaxer and the anti-convulsion, and this is called the atizolam. So these are the different generic name of these compounds that these are the formulas which are present in these drugs. So the drugs are then sold by the different brand names, but you always see that there is a particular formula, and that is basically that compound or the active ingredient, and we also call it API, right, which is active pharmaceutical entity or the ingredient which is available in that drug. So you can see that how if you just look carefully, so there is a not very major difference in the structure. But as a chemist, as an organic chemist, we do realize that even just the methyl group substitution, just the rotation of the double bond, it has a significant impact on the structure. It changed the whole of the identity of the molecule. So now you can see that even it has the chlorophenyl substitution on these sides. There is the 7-membered ring in all of these ones. Then there's a phenyl and the chloro. There is a thiophene kind of moiety there, but there is only the minor changes in the structure, and they are using for the different drugs. So that's how the molecules behave. Sometimes the molecules are also able to treat 2 types of the targets or the proteins. Sometimes they are very specific that they are only able to work on one particular kind of molecule or the protein. So now coming to what the acetylcholinesterase is. So the acetylcholinesterase is one of the enzymes which we do have in our body system, and this is basically responsible for the acetylcholine hydrolysis. And whenever the drugs are basically synthesized and they are discovered for the treatment of the Alzheimer's diseases, so they are basically the acetylcholinesterase inhibitor. I will tell you that why we are saying this. So in the acetylcholinesterase, there is another similar kind of enzyme, which is the butyrylcholinesterase enzyme, but that's present in the plasma, so that doesn't have that much of the importance when we discuss about the neurotransmitter acetylcholine and its catalysis or its particular hydrolysis. So this is basically the acetylcholinesterase enzyme. So let me give you the next slide. So the acetylcholine is an organic chemical that function in the brain and body of many types of animals, including human, as a neurotransmitter. So it's one of the very significant neurotransmitter, a chemical released by nerve cells to send signal to other cells. Its name is derived from its chemical structure. It is an ester of acetic acid and the choline. So the choline, chemically, it is the 2-hydroxy NN trimethyl ethane aminium compound. And let me show you the structure. So this is the acetic acid. This is the choline, which is the N-trimethyl ethanaminium compound, because right now this is in the quaternary structure, because the nitrogen is making the 4 bonds through its lone pair of electrons as well. So now the ester of this acetic acid and the choline, because the choline has the hydroxyl group And we know from our basic organic chemistry, so whenever we react the carboxylic acid with the alcohol, we get the ester. So this is the one of the ester, which is the acetylcholine, right? So that acetylcholine release as a neurotransmitter in our brain system. So once it delivers its message from one nerve to another nerve, so it has to stop. So there is an enzyme which is called the acetylcholinesterase enzyme, and that work on this acetylcholine. So it basically hydrolyzes it back into the acetic acid and the choline. So there is not overexpression, because that will also create the paralysis, that will also create the abnormality. If there is the continuous signal, which we really do not need. So you might have experienced some of the people who have uncontrolled reflex action, because there are the many of the such kind of chemicals or the neurotransmitter which are constantly releasing. So this kind of activity, what the nature had designed, that after delivering its message, it has to stop, it has to hydrolyze back, and then when there will be the new message, so that will come back, and from the receptors, it will get to the next nerve. Now, for the Alzheimer, so this is basically how it happens in our brain system. So every nerve, we have the neurons in our brain. So every neuron has one of the terminal where these two nerves are separating, and there is no such passage or the tunnel that it actually transfer from one nerve to another. So this is whole through the electronic signals. So these are through the neurotransmitters. So the one nerve then send this acetylcholine to the second nerve. So there are some of the receptors which capture these ones, and then they receive the message, and then they deliver the acetylcholine to the acetylcholinesterase enzyme. So this enzyme then cleave back, hydrolyze this acetylcholine into the acetic acid and the choline. So now there is a message which has delivered. So now we do not need acetylcholine, much of the acetylcholine release. So it has to get back. So whenever there is the Alzheimer or dementia or these kind of the diseases, so they already do not have that control. So they are facing the loss in their memories and the control on their nerve system. So that's why this is important, that it has to continue, because they are already short in that one. So then for that, this acetylcholinesterase enzyme activity has to stop. It means we wanted to stop the hydrolysis of the acetylcholine. We want to prolong that one. So they get that message because they are already facing the shortness of this one. So now we have to create such kind of compound which can bind with the acetylcholinesterase enzyme, and it just mimic, it just deceive it in that way that the acetylcholinesterase enzyme look it as the acetylcholine, but this is not the acetylcholine, this is our drug, and this is binding with the acetylcholinesterase enzyme. So this enzyme is not now able to directly capture the acetylcholine for its hydrolysis. So something we are giving as the alternative, and some of the you can say the disguised partner, that it will make the bonding with that one, but it is not working actually on the actual compound which we wanted to release it further. So that is first thing which in the first slide I have mentioned you. This is the target identification. So for treating the Alzheimer, we identify that the acetylcholinesterase enzyme is our target, and now we have to design the drugs which can work on this enzyme, and then they can inhibit their activity. So for the synthesis, now the part is how the organic chemists do design any of the scheme and how they finally synthesize the compound. So through the research, through the computational tools and the literature review, so we see that this kind of structure of our compound should be there. Let me highlight this for you so you can see that this was our target molecule. That in our molecule, which we wanted to propose as a drug contender for treating the Alzheimer, this should be the functionality in our molecule. Now we got the structure. Now our job, now this is our challenge, that how we will synthesize in our lab, from which we should start. So the first thing, as a chemist, the approach is that you have to utilize your organic synthetic approach, that which bond are easily breakable, which functional group conversions and the modification I can do. Now, in this time, we also have some of the tools which also help us. And in most of the countries, these are not obviously freely available. One of the thing is the SciFinder. There are other organic portals. So you can also take the help from these one, that you draw your molecule, and they will give you some of the suggestions that could be the synthetic step to synthesize these compounds. But again, you have to see that which is economically favorable. You can't go to the very expensive compounds that you just paying the $700 for the 1 mg of the compound. So you have to utilize, because as an organic chemist, we can synthesize these compounds in our lab. So we always start from the less expensive or the cheaper and the more available compound, and then we can do the modification. So for synthesizing this compound, rather to get any of the triazole from the vendor, so that might be very expensive, and we might not be able to fuse with the phenyl ring directly, or even if we get that was too expensive. So what we did is that we see that, okay, so this bond is easy to cleave. Let me do that again for you. So we can introduce, for example, this moiety we can bring from somewhere. This we can do later. We can add this side to that one, and then we should have some of the thing which can make this cyclization of the triazole, and there should be this substitution with that one. So by approaching our organic knowledge and going backward, this is called retrosynthesis. Retrosynthesis means you do the reverse engineering, that rather to start what we usually do in any of the chemical equation or any of the reaction, we usually do a forward a pattern, that, okay, this is the first reactant, we are mixing with the second, and then we are doing. But for the retrosynthesis, what we do is that we have our compound. So firstly, we analyze that which bonds are easy to make, or which functional group transformation is easy to make. So we will design our scheme. So by looking at this structure, we design our scheme in this way, that what would be our route, how we will synthesize that one. So we started from the simple carboxylic acid, and from the carboxylic acid, that was easier for us to easily transform into the ester. One of the most common esterification reaction is a fission esterification, but you can use any other methods as well. So in the presence of the concentrated sulfuric acid, using the ethanol as a solvent, and under the deflux condition, we transform our carboxylic acid into the ester. What was the reason? The reason was this, because we wanted this carbon, right? And we wanted to have introduce this nitrogen. And for this, we need some of the functionality which can easily cleave, because if we do with the acid, so that was not possible chemically, right? That whenever we do some of the harsh condition, this hydrogen will get out, right? So we cannot able to get the nitrogen compounds. So what we did, then we transform it into the ester. So in the ester, this methoxy or the ethoxy, depending on what kind of alcohol you're using. So we use ethanol, so that's why there was the ethyl group which has added to that one. So this ethoxy is always a good leaving group. making this carbonyl as the electrophilic one. So when we treat it with the hydrogen hydrated, which is NH2NH2, and it has one water molecule into that one, in the presence of the methanol, so this was easily break, and we get NH and NH2. Now we have acquired one portion of our molecule that we will get the carbon from this carbonyl, we will get the nitrogen and the nitrogen from these ones. So for this, we wanted another nitrogen, right? And the carbon and the sulfur at the end. So what we did, we used the 1-chloro-4-isothiocyanate benzene for this. And then this reaction was also happen in the methanol in the presence of the reflux. So the first step was this to reflux that one. So in the first step, we get this uncyclized moiety, and then when we treated it with the basic condition, and for the potassium hydroxide is a very nice base which helps for the cyclization of any of the molecule, because this is the basic pH dependent reaction. So when we treat it with the 10% potassium hydroxide aqueous solution, so what we get is that this uncyclized molecule gets cyclized, and we get the 1,2,4-triazole with the thiol moiety at the end. Now, what is the importance of this thiol at the end? Because we wanted the sulfur, and second thing was that we want this moiety, which is acetamide, at the end of this molecule. If we do not have such kind of acidic hydrogen at the end, so we can't do the functionalization, or we can't do the derivatives of this one. So this is important that our parent compounds do have such kind of group where we can just remove the hydrogen or any of the good leaving group, and we can introduce the rest of the functionality into our molecule. So for this acetamide, what we did is we utilized the different, this was another kind of reaction, and we prepared the series of the acetamides in our lab. So we used the bromoacetyl bromide, and we reacted it with the different kinds of amines. Practically, we more focused on the aromatic amines, for example, the aniline. And when we treat it with the bromoacetyl bromide, so that give us the acetamide, and still there is a one of the bromine. So that's why it can easily go with this thiol side, because this hydrogen and the bromine will leave, they will make a bond and they will leave that one. So this bromine is basically kicking off this hydrogen and taking up with itself, and the rest of the alkyl group will attach to this sulfur. So we did this parent compound series and the derivatives in the two ways. In one, we did the acetamide functionality, and in other approach, we just changed the alkyl, like we substituted this alkyl with any of the alkyl halide. We started from the methyl up to the 7 carbon chain alkyl groups to see the behavior, that either there is an impact if we increase the chain length of the hydrocarbon, what is the impact of the acetamide derivatives of these 1,2,4-triazole. So once we created this 21 or the 22 compound series, then we did their by characterization firstly, because that is very important, that our compound which we are aiming has been synthesized. So I'm representing just one of the compound spectra to you, just to give you the idea that after the synthesis, you have to confirm the structure of your compound. So we did the proton NMR, we did the carbon-13 NMR as well. Why? Because if you just look at the structure, so the proton NMR was not enough, because this heterocyclic moiety do not have any of the hydrogen. So for confirming its structure, we need the carbon-13, because the carbon-13 is giving us the environment of the carbon, and then we can confirm any of that moiety which has a carbon but do not have the hydrogen there. Then after the characterization, the next thing was to conduct their biological analysis. So for this, we did the enzyme inhibition assay, and the protocol of this one was that enzyme, we set up a buffer for maintaining the pH, and then the or molecule which it will work as an inhibitor. So we mix the enzyme, we add the buffer to maintain the pH, because we know our body blood system has the pH around the 6.4 to the 7. So we have to keep the pH optimum, so to see the true results of our compounds in the cellular pH. So then we use a 96 well plate. The substrate was there, which is our protein, right? And then we incubated this one. Then we measured absorbance. So with the concentration of our compound, so what was the absorption and what is the inhibition? So by absorbing the by measuring the absorbance, we indirectly looking at inhibition of our compound towards that enzyme activity. So from these biological assay, what was the result of our compounds? So this was the whole of that series of our compounds. So here I'm representing the 7 of these ones. This was the hydrazide, then there was the triazole, then these were the 5 different derivatives of these series. Now the neostigmine is one of the marketed drug already available for this enzyme activity, because this is also to compare your result with the one of the standard drug, to see that how is it good for that one, or if it is not of that standard, so then if there is already available something, so you will then go for the further modification which can enhance the results. So we also do the blank when we are not using any of the inhibitor. So what was the activity of the blank at that moment? So now if you just look at this one, so our HABR5 was among this series of the compounds which show the 98% of the inhibition. And if you just see the new segment, which shows the 99.4% inhibition. So this is quite comparable. And then when you examine the IC50 value, so the IC50 value is basically stands for the inhibitory concentration, which is required for the inhibition of the 50% of that enzyme activity. So the IC50 value, minimum the IC50 value of your compound, it means the minimum concentration you are using of your compound, and that is showing the potential, that is giving the activity. So if your compound needs in a very high amount, so that means that you need to think about, and you have to modify your compound, because this is not ideal. We can't go for the very high doses. So it is always ideal that if you get the IC50 value at the lower amount. So then after that one, when you compare the ACHE studies, and these are the bar charts to give you the idea. So here you can see the IC50 value of HABR, which means the minimum concentration as compared to the neostigmine, right? So here you can see just less than the 20, right? If we need HBR4, while the neostigmine, which is already available, that has to be around the 100. So this is much far in amount to show the result, while our HABR is showing much of the greater result, and it is equal kind of inhibition as well. So now the thing was that from our series, this concentration, we also did the dose dependent analysis as well. We get that from the series HABR5 is giving promising result, and HABR1 was also giving some of the good results. So we compare that if we do their dose response study. Dose response means once we identify that what is the maximum amount they are giving the maximum inhibition, then we have to go do that one. Then at what dose, if we starting from the, for example, the 10 millimole, then 20, then 30, then 50. So what is the trend in the dose, and at what dose this is giving the maximum of the inhibition and where it turned back. Because many of the molecules, when you exceed certain amount, they just get deactivated for the protein, and then either and sometimes they are always in a linear way, that they, whenever you are increasing the amount, so they will increase the percentage inhibition. So when we did the concentration of the compound, so the first we use the we also use starting from the 0.7 to 1, then 2, 5, 11, 22, and 45 millimoles of that one. So here that was a trend we see in HABR, we see HABR5. So these are the IC50 values of these ones. And this is the percent inhibition of that one. So here you can see that the percent inhibition with respect to the blank of the HABR, if you compare, so this is giving the 98.8% with the 0.001 for IC50 at the 45, while HABR is having the IC50 value 0.02, and it is just giving us the 78.57% inhibition. So it means HABR5 is the more promising compound we synthesize in our series of the compounds. So then we also compare these dose dependent charts in the bar chart. So this is the same kind of results. We just translated into the bar chart to make it more visual, that what is the trend of the HABR5, right? So if you, I just wanted to show you the structure. So HABR has this structure. There is a phenyl group, there is a triazole ring, there is the 4-chloro substitution, and we just did the reaction of the aniline with the bromoacetyl bromide to make this bromoacetamide derivative. So this is HABR1. And if we compare the HABR4, which was the HABR5, which is the more promising one, so you can see the rest of the compound is same. So the only difference is this one, that it has a nitro and it has a chloro substitution. So because of these substitutions, it has changed its activity, and it has a much of the enhanced activity as compared to the HABR1. So the point is this one, that whenever the chemists do not get their required compound efficiency, so they go back and they then add some of change the different functionalities and the substitution, and they see. So this is all is the hit and trial method, because you synthesize the different range of the compounds, and then you do their assays, bioassays, biological activities analysis, and then you find out that, okay, that is the result. So now this is one of the picture from our computational results. So the computational, as I said in the start, is also used for the designing. It gives you the prediction that you should synthesize such type of compound, which should have these major functionalities. So now it has improved the productivity of the pharmaceutical industries, the pharmacists, the medicinal chemists in that way, because when we were not aware with the computational tools, there were number of compounds you synthesize, and everything demands your time and the cost. So now, due to the resources of the computational, this is much of the better, that you are not wasting your time for the inactive compounds. So you are not creating all of the compounds in the lab. So you just go for the hit compounds, which are coming through the sum of the computational results. Okay, that compound should have these kind of activities. So that might be the more potential. So then you translate into the lab. So you, rather to synthesize 100 of the compounds, now you just get the top 10 from the computational tools and you synthesize that one. And then you go further for the biological analysis. So if you just look at this picture, we know that all of the proteins are made up of amino acids. So the primary structure for protein is the amino acid sequence. So these PHETYR are the 3-letter abbreviations of each of the amino acids. And this is our molecule, which we just have shown. And you can see these dotted lines are showing the interactions of the compound different substitutions with the different amino acid residues. So you can see how actively it is interact with the amino acid residue. So the binding is very important for your compounds with that protein to get bound with that one and to do the enzymatic activity by the enzymes. So that's why then that could be the effective compound for treating any of the disease and to bind with any of the target. Now, at the end, these are the references. Some of our papers we publish in the different journals as well, and how we synthesize and what are some of the other papers I have cited for the introduction. At the end, I would introduce my research group to you. So this is my current, and this is a badge 4. I have the MPhil students, I have the undergrad student as well. So right now we just have only one guy as an undergrad, but these are working on the different projects. So some are working on the HCV and some are working on the anti-cancer, and one of the group is working on the anti-leishmanial drugs, anti-leishmanial disease. This is our campus and this is our science block building in the Pharmacological College, Lahore, Pakistan. And at the end, I really thankful to the organizers, the University Jambi, and representing my university, the Pharmacological College University, for this wonderful collaboration and to interact with all of you guys across the other countries. And hopefully we will see the more collaborative activities in the future as well. And at the end, thank you so much. And I really appreciate if you have any questions regarding my presentation or generally about the discussion of the drug discovery and the trials. Thank you so much.
Thank you so much to Professor Khalid for the very informative presentation.
Thank you. I'm sorry I take much of the time, maybe because
listen to. Yes, of course. So to our audience, please, if you have any question to
I think there's a distortion in the sound.
Khalid.
Yeah, there are some of the
Any
questions.
question from audience?
Yes, they are the two hands raised. Yeah.
That's right. I think it has this problem.
I'm sorry,
Okay.
ma'am. I have a connection problem. Okay. So we have question to Muhammad Furqan Novrian, please.
Yeah, thank you for this honorable opportunity, Mr. Hira Khalid. And I think it's thank to Dr. Hira Khalid for the opportunity and us the big and amazing knowledge about the inhibitors cholinesterase. The thing that I want to ask about the questions, is that possible to find any peptides or the essentials plan from the essential plan, like an essential amino acid or a peptide to use as an cholinesterase inhibitors? As we know, the acetylcholine is a neurotransmitter that we need for our brains, our human brains or neuron systems to controls cognitive functions, and it also need by Alzheimer's diseases. So what is something we can say we considered if we want to make inhibitors of cholinesterase, or it is possible if we use a peptide or the essentials plant or extract from a model from the environment plant, we can use it as an inhibitor cholinesterase, and how's the mechanisms to working from the inhibitors cholinesterase can be increasing a rate of the acetylcholine in our brains, how the mechanism and how the progress to acetylcholine in our brain is increasing immediately. I think just that from me. Thank you. Dr. Hira Khalid, is glad to see you here. Yeah, just like that. Thank you.
Thank you, Furkan, for asking a wonderful question. But I must say this is not a single question, so there are many of the things in a single question. So let me address one by one. So the first thing is, you are very rightly, because there are many of the people who are working on which is called the peptomimic compounds, right? So they are synthesizing and they are working the peptide base of the compounds, and they are not only utilizing these for the Alzheimer or the acetylcholinesterase, but there are some of the or for the drugs for other diseases as well. So yes, you can work on it, and we can find for that solution. And there are many of the people who are working on the peptido-based drug chemistry as well. The second thing is, this is something, because the acetylcholinesterase and the Alzheimer and the dementia, we say, so this is more linked with majorly with the aging. When you age, so there are the decrease in the release of the acetylcholine. So that's why this is compromising the many of the nervous system issues. And the second thing is, there are many of the habits and which are also the environmental factors which are also influencing that one, the radiations, the medications which are also damage the acetylcholinesterase and the nervous system. So there are many of the opportunities to explore the plant-based compounds, as you said. So for example, because I also have the idea, but I really interested to start that one, because from if you know, I don't know in Indonesia what we call it. So this is the you can say the pumpkin, and this is a zucchini kind of vegetable, right? So this has a very good potential for the activating the neurotransmission. But when we work on the plant, so this is more of the time demanding thing, because you have to go for the extraction, you have to extract the active compounds, and you have to go for the total synthesis. So then to analyze that what is the behavior, either that is the collectively, because when we use any of the plant-based extracts, so this is called the synergistic activity, because there are a number of other ingredients which are helping for showing some of the biological potential. And when we isolate any of the single kind of molecule, so this is one of the active molecules, so then we have to see that either that is also responsible for contributing to that activity or not. But there is again a lot of room to work on that one. Thanks for asking this. Yeah.
Yeah, thank you, Dr. Hira Khalid. That was great. Thank you.
Thank you.
Thank you, Furkan. Anybody else have this question?
Maybe I want to ask to Dr. Hira.
Yeah, sure.
Hello, Dr. We meet again. Let me introduce myself again. My name is Rahul Bikranza, and I want to ask to you that they have diagrams and the presentation before. They why does a concentration of 40 have a high percentage inhibitor value that a concentration of 2020 2020 there is a drastic decrease in the presentation is inhibitor. Yes, this one.
Okay.
In
So your
20.
your question is what was your question is that we have it is decreasing with the different amount. Can you just say this if I get your question rightly? So your question is that with the 45 we are having the 98 of the percentage, right? And you are asking that why this is going down with the different percentage. That might be your question.
Yes, Dr. Hira. Yes.
Okay. Yeah. So basically, if we just focus on one of that compound, HABr5, right? Let's say take this as an example. So this concentration of the compound means that we wanted to set up that if we take it in this little amount, so what is the percent inhibition with respect to the blank? The blank is when we are not adding or inhibitor. So it means there is no inhibition activity. So when we use this much of the concentration of our compound of the HBr, right? So if you just look at this one, so this showed the IC50 value of 0.0941, right? And the percentage was the 20% inhibition. Like at this minimum of the amount, this was just showing the 20% inhibition, means the only 20% of the enzyme was inhibited for its enzyme activity. When we increase the activity, another important factor is, if you just look carefully, so the IC50 value, so this column is basically representing the IC50 value. So the IC50 value of this at this amount of the compound, which is far less than the 45. So the IC50 value is the here is the 0.09, right? And when we increase that one up to the 45, so here you can see this is the 98.82% of inhibition, and the IC50 value was the 0.0014. It means that this little amount of the HABr5 rate, right, with the concentration of the compound, so that is showing that amount of the percentage. We always go where is the maximum of the inhibition, and we also have to focus on the minimum inhibitory concentration. So sometimes we have the compounds, for example, if our compound is showing the 98% of the inhibition, and in other of the dose or other of the compound is showing the 78% of the inhibition, but the IC50 value of the other compound, which is showing the 78%, is far much less. So we always prefer that compound which has the least of the IC50 value. So that's why if you just look at this one, so at this concentration of the solution, so here the active compound minimum concentration to inhibit the 50% of the enzyme activity was just the 0.0014, right? So that's why this is our ideal score, that we are not compromising the patient health, because if we are giving much of the dose to that patient, so that will be harder to excrete. So every drug has to excrete out after doing its work from the body, because if you are accumulating all of these compounds and the chemicals within the body, so that these are damaging, they have this other side effects on the other organs as well. So I hope that answers your question.
Okay, Dr. Hira, thank you.
Yeah, welcome.
Thank you so much. to Professor Khalid. Anybody else want to discuss or ask a question about the presentation that we just listened?
May I, Mr. Irhash?
Yes,
Thank
sir.
you, Prof. Hira's amazing speech today, talk today. I would like to ask about the current progress of all of the compound that you have synthesized, like have been used the in vivo assay for verified whether the compound is active in the host, like the mouse or the other animals that we can use, and how then the small compound drug can deliver to neurotransmitter and its may affect for the activity.
Yeah, thank you so much, Andra, for asking this one. So we did their biological assay, and currently their animal testing is in a process. So yeah, so we are working on that one. Yeah. So then we will be able to, because that dose was on the base of our in vitro studies, but now because we get even the good results, so now we have transferred to the animal testing. But animal testing taking some time because we have to collaborate with the pharmacy, and they always take your compounds, and we are just waiting for their response. So yeah, these are in the process.
Okay, but how the drug then you can inject is by the solution, like they inject to mice or
okay. So yeah, so there are the different ways because they're also dependent on the drug. If it is water soluble, so we may provide on the basis of solution. Sometimes we also do that we mix it with their feed. So we mix it with the feed with some of the dosage, and they take it, and then we experience their responses after the day, 24 hours, and 72 hours, and then the weeks and the weeks. So either there is any of the response of the side effects, they get unconscious, nauseous, or something. So then these also giving us the response for their activity, because firstly, for the animal testing, we induce something which we wanted to inhibit, and then we provide that inhibitor or molecule in the form of either the solution or we give them through the syringe, just like you give the milk to the babies or the something. So we feed in that way, or otherwise we mix it with the normal feed where they eat that one. So then we test their responses for the toxicity, and for the neurotransmitter, then we have to unfortunately do that some kind of dissections as well for seeing that responses afterward, that how that was the inhibition, if we have to measure that in the signaling, for example, and if we are treating some of the heart. So yeah, so then we do some kind of dissections and extract these cell lines to see that thing. Yeah.
Okay. Thank you.
Thank you so much for Mr. Indra for the question, and also Professor Khalid for the answer. Okay, I think for last question, I would like to place the audience if there is any. Okay, I think there are no questions. For today, I would like to give some comment to the presentation. I think the presentation is very informative today. We got so many valuable knowledge. First, we are hearing about the process of when I was in the US, that this process is, yes, this is a very long process. It takes years. Like we just get information from Professor Khalid, of course. And also my professor said that for drug discovery, actually, we have to prepare a lot of money. she said. And yeah, that's true. We can see like how many budget and expenses for the even from the preclinical trial to clinical trial, there are we need a lot of budget for that. And also we already witnessing the strategy that used by Professor Khalid in her lab for the synthesis of the triazole compound. That is, I really got amazed by the approach, and I think that is very smart approach in a term of forming the triazole ring. And then also we see how Professor Khalid in the lab characterized the compound, and also do the bioactivity assay. And yeah, I think like we already got the whole picture of how drug discovery conducted. And thank you very much once again, Professor Khalid, for
Thank you
this
so much.
very amazing information to us, and hopefully it will stimulate students to get attracted to this field of chemistry. Okay, thank you so much.
Thank you so much, Mohammed. I just wanted to ask, say something at the end, because this approach is giving also the idea that this is not
okay, so
on.
I got information from the committee in here, ma'am. So I will give you a certificate, kind of certificate.
Okay, thank you.
Before we
Thank you so much. I really overwhelmed.
the event today.
Thank you.
So this is the electronic certificate.
Thank you. So I hope I will get that in an email.
Okay.
Right? Yeah. Thank you so much. So thank you, Mohammed, and thank you, Indra, for all of your communication and this invitation. And wish you all the best for your department and the university as well. And thank you, Rahul, for being a very active student as well. So it was nice, and I think she was Alina or Alvina. I'm sorry if I'm not pronouncing her name. So thanks for moderating this session, and thank you, Mohammed, for moderating this session so nicely. And thank you so much for the opportunity. It was a pleasure.
Thank you, Prof. Thank you.
Thank you.